welcome to the webinar on automated methylation sequence analysis using Mutation Surveyor software by SoftGenetics. My name is Brent DiGiorgio, and I will be the presenter for this webinar. Methylation of DNA at cytosine positions of CpG dinucleotides is associated with developmental factors such as X chromosome inactivation, genomic imprinting, and various diseases such as cancer. Bisulfite conversion has become the most widely used technique for studying DNA methylation, which chemically converts non-methylated cytosines to uracil while leaving methylated cytosines unaffected. Nucleotide sequencing of bisulfite-treated DNA will display the unmethylated cytosines as thymine, while methylated cytosine positions appear as cytosine in the sequenced DNA. Mutation Surveyor offers a unique function that simplifies the analysis of bisulfite-treated DNA by displaying methylated and unmethylated cytosines on the amplified sequence of the send strand. This image displays an outline of bisulfite sequencing of genomic DNA. After denaturation and treatment of the double-stranded DNA, the blue cytosines that are unmethylated are converted to uracil, and the methylated cytosines depicted in red text remain unchanged. The resulting fragments are then amplified and sequenced, and the unmethylated positions that were converted to uracil are displayed in the amplified strand as thymine. Mutation Surveyor utilizes a patented physical trace-to-trace comparison technology to detect nucleotide changes in methylated sequences. By using the program's methylation function, Mutation Surveyor displays methylated and unmethylated cytosine positions in the reference trace. Cytosines within CpG islands are marked as methylated in the reference trace, and cytosines outside of CpG islands are marked as thymine to indicate conversion. Bisulfite-treated sample files are then physically compared to the reference sequence in order to detect methylated positions. We can begin the webinar by loading files into the program. Mutation Surveyor displays methylation information in the reference sequence by utilizing a GenBank file to construct synthetic reference traces. For this reason, the GenBank file must be in the forward strand orientation so the CpG sites are well defined, and the reverse complement sequence must be converted from the GenBank file. This can be done through the NCBI website or through Mutation Surveyor's Advanced GenBank File Editor. For more information on this process, please refer to the webinar on downloading GenBank files. After loading your GenBank sequence file, you can load your sample files. The reference pane should be left blank, as the program synthetically constructs reference files for mutation detection. After loading your files, click OK, and you will be prompted to reverse complement your sequence. Select No to All, and you will then be able to adjust your mutation project settings. Navigate to the Process Setting options at the top of the page and click on the Others tab. Check the box at the bottom of the tab that says Methylation to use the methylation function of the software. The user can then select which cytosines in the reference trace will be displayed as thymines. It is recommended that users select the auto-methylation function, which will display any cytosine positions that are not next to a guanine as being methylated. When your project settings have been confirmed, click on the Run icon at the top of the screen to begin the analysis. The main mutation report displays any variants that were detected in the analysis. These positions indicate sites of partial methylation as well as sites of heterozygous mutations. You can double-click on a position within the mutation report to open it in the graphic analysis display, or you can double-click on a trace in the trace pane to open a view of the entire trace. If you zoom in on the nucleotide positions within the trace, you will see how Mutation Surveyor identifies sites of methylation within the sample files. Thymine positions within the reference file that have a blue bar placed over the letter represent unmethylated cytosine positions that were converted to uracil. We can see from the GenBank sequence in the nucleotide sequence pane that these positions were originally cytosines. However, because these positions are not located next to a guanine nucleotide, they are converted to uracil and appear as thymine in the reference trace. In this sample, we can see that there are many cytosine positions that were converted to thymine, indicating that the positions are unmethylated. We can also see from this example that the cytosine positions located within CpG dinucleotides remain marked as C in the reference trace, indicating that these positions are possible sites of methylation. The sample file displays cytosine nucleotides at these positions, indicating the presence of methylation. The positions with spikes in the mutation electropherogram represent sites of partial methylation or heterozygous mutation. The variants displayed in this sample indicate positions with a mixture of cytosine and thymine nucleotides and are indicative of partial methylation. We can now cover some of the reporting tools available for methylation analysis. With the methylation option selected, a methylation report is available by going to Reports, Customer Reports, and clicking on the Methylation Report option. This report lists the positions within the sample trace relative to the GenBank file. Cytosine positions within CpG islands are marked with an M for methylated or U for unmethylated, and cytosine positions outside of CpG dinucleotides are marked with an S for successful conversion to uracil or an I for incomplete conversion to uracil. The report can hyperlink to the graphic analysis display, and double-clicking on a call within the report will open that position. If you wish to save the report, you may copy it to the clipboard or save it as a text file by clicking the Save icon. The pattern of methylation can also be viewed in a graphical report by clicking the graphic display icon. This report displays the pattern of methylated and unmethylated cytosines along the sample files used in the analysis. Red tick marks indicate methylated cytosines, and blue tick marks indicate unmethylated cytosines. This report also includes a methylation ratio and conversion ratio at the bottom. These ratios indicate the percentage of methylated and unmethylated cytosines both inside and outside of CpG islands. Another useful tool for methylation analysis is the Mutation Quantifier tool, which can be accessed by clicking the Mutation Quantifier icon at the top of the graphic analysis display. This option enables you to quantify the allele ratios at specific locations within the genome, and it can also be used with the methylation function to quantify the ratios of methylation at specific positions. The tool provides a simplified allele ratio, which displays percent normal and the percent mutation, and a standard allele ratio, which evaluates the drop of the normal allele and gain of the mutant allele. For more information on the differences between these two, please view the Mutation Quantifier webinar. To display the function of the tool with methylation settings, I will choose Simplified Allele Ratio Settings. The Simplified Allele Ratio report displays the samples within mutation calls and lists the percentage of the normal and mutant alleles in the last two columns. Those positions with partial methylation are indicated in the report, as well as the positions that contain heterozygous SNPs. You can click on any call within the report to open the position in an electropherogram window. We can see from this partially methylated position that it has a thymine contribution of 79.93% and a cytosine contribution of 20.07%. If you wish to save the details within this report, simply click on the Save icon at the top of the page. This concludes the webinar on methylation sequence analysis using Mutation Surveyor software. If you are interested in learning more about the many features of the software, please view our other webinars. For more information, or to receive a free 35-day trial of Mutation Surveyor and other Soft Genetics products, please visit softgenetics.com or send an email to info@softgenetics.com. For technical questions about the software, please send an email to tech_support@softgenetics.com. Thank you for joining me in this webinar.